Hello everyone. Today we will start on a journey to understand not just the 12 cranial nerves themselves, but the fundamental way their functions are organized within the brainstem. That is through their functional components and the nuclei they arise from or terminate in. This is a crucial topic for your understanding of neuroanatomy and will form a vital foundation for your future in clinical medicine. As you can see, we have a visual representation of the brainstem, which is the central hub for the majority of these cranial nerve nuclei. Let's begin our exploration of these essential structures. Before we dive in, it is important to understand what we aim to achieve by the end of this session. By the time we conclude, you should be able to confidently first identify and differentiate between the various functional components that make up each cranial nerve. Think of these functional components as the different types of fibers within each nerve, each with a specific job. Second, you will be able to pinpoint the location of the specific nerve cell bodies or nuclei within the brainstem that are responsible for each of these functions. It's like knowing where the control centers for each function are located. The third, we will explore how these nuclei are not scattered randomly but are organized into longitudinal columns based on their function. This organization is the key to understand the brainstem structure. Fourth, you will be able to connect these functional components and their respective nuclei to the actual roles each cranial nerve plays in the body, what movements they control, what sensations they carry, and so on. And finally, you will begin to appreciate the clinical relevance of this knowledge, understanding how damage to specific nuclei or functional components can lead to distinct neurological deficits. This understanding is crucial for diagnosing and treating neurological conditions. Let's start by familiarizing ourselves with the stars of our video, the 12 cranial nerves. As we can see listed here, they are numbered 1 to 12 using Roman numerals and have specific names that often hint at their primary function or distribution. For example, the olfactory nerve is involved in smell and the hypoglossal nerve affects tongue movement. It's important to remember that majority of these nerves, specifically cranial nerves 3 to 12, emerge directly from the brainstem, which is the crucial structure connecting the cerebrum or the main part of the brain and the spinal cord. Cranial nerves 1, that's the olfactory nerve, and 2, the optic nerve. They have unique origins and pathways that we will touch upon briefly, noting that they are more accurately considered outgrowths of the forebrain. Each of these 12 nerves performs distinct roles in controlling motor functions, relaying sensory information, or influencing autonomic activities. These diverse functions are not arbitrary. They are a direct consequence of the specific types of nerve fibers that make up each nerve, which we call the functional columns, and the specific groups of nerve cell bodies or nuclei they are connected to within the brainstem. A key concept we will explore today is that these nuclei are not haphazardly arranged but are organized in a longitudinal fashion within the brainstem, forming what we call the functional columns. This organization reflects both their developmental origins and their shared functional characteristics, making it easier to understand their functions and relationships. The cranial nerves are not simply single entities. They are composed of different types of nerve fibers, each carrying a specific type of information. These are known as functional components. To truly understand the function of each cranial nerve, we need to look deep into their fundamental building blocks, the functional columns. Think of each cranial nerve as a complex cable containing different types of wires, which is shown schematically, and each wire carries a specific kind of signal. These signals represent different types of motor or sensory information. We categorize these wires or nerve fibers based on their origin, the destination, and the type of information they carry. Let's go through each of these functional components in detail. We will look into the efferent components now. First, we have general somatic efferent, abbreviated as GSE. General here refers to widespread. Somatic refers to body's framework, that is the muscles and skin. And efferent means carrying signals away from the brain, so motor signals. These are motor fibers that control skeletal muscles derived from embryonic somites. Somites are early developmental structures that give rise to much of our musculoskeletal system. Think of the muscles that move your eyes, allowing you to track objects, or the muscles that move your tongue, enabling you to speak or swallow. These are typically innervated by GSC fibers. The next is branchial motor, sometimes also called as special visceral efferent or SVE. These are motor fibers that innervate muscles derived from the pharyngeal or branchial arches during embryonic development. These arches give rise to structures in the head and neck. This includes muscles of mastication, which you use for chewing, the muscles of facial expression, allowing you to smile, frown, and convey emotions, and the muscles of the pharynx and larynx, which are crucial for swallowing and speech. The term special visceral highlights that these muscles, while controlling movement, have a unique developmental origin related to the viscera. Then we have general visceral efferent or GVE. Here, visceral refers to internal organs. These are also motor fibers, but they innervate smooth muscle, like the muscles in your digestive tract, cardiac muscle, the muscle of your heart, and glands. These are part of the autonomic nervous system, specifically the parasympathetic division. So, these fibers, these GVE fibers, control involuntary actions like constricting your pupils in response to light, stimulating salivation for digestion, slowing down your heart rate, and promoting digestion. Moving to sensory components, the general visceral afferent, here it is, or the GVA. These fibers carry sensory information from our internal organs or viscera. This includes sensations like distension or stretching of organs, pain from internal organs, and changes in blood pressure or blood chemistry. For example, GVA fibers tell you when you are full after eating or when you are experiencing abdominal discomfort. The special visceral afferent or SVA, these fibers carry the special senses of taste. Again, special and visceral highlight the unique nature of these senses. It's important to note that while smell is a special visceral sense, its primary pathway does not directly involve brainstem nuclei in the same way as taste. The next is general somatic afferent or GSA. These fibers carry general sensory information from the skin, muscles, and joints of the head and face. This includes sensations like touch, pressure, pain, temperature, and proprioception, which is your sense of where your head and face are in space. So, if someone touches your cheek or you feel a headache, that information is carried by GSA fibers. Finally, the special somatic afferent or SSA, these fibers are responsible for our special senses of vision, hearing, and balance. Special here indicates senses with the dedicated sensory organs. Understanding these functional components is key because each cranial nerve is a unique combination of one or more of these fiber types, and it's these components that dictate the specific function of that particular nerve. Now that we understand the different types of nerve fibers or functional components, let's see how the nerve cell bodies or nuclei associated with these functions are organized within the brainstem. This slide shows a simplified representation of cross sections at different levels of the brainstem, the pons and the midbrain. The brainstem, as we have already discussed, is the connection between the cerebrum and the spinal cord, and it is the origin point of most cranial nerves. You have to notice that the nuclei, which are clusters of neuron cell bodies, are not scattered randomly but are arranged in longitudinal columns that run along the length of the brainstem. These are known as the functional columns. In this diagram, these are the motor nuclei and these are the sensory nuclei. Generally, we can observe a pattern here. The nuclei associated with somatic efferent functions, those controlling skeletal muscles derived from somites, they are located most medially. This medial position is a general rule for motor nuclei controlling skeletal muscle. And further laterally, we will identify the nuclei associated with visceral efferent functions controlling the smooth muscle, cardiac muscle, and glands. These are positioned more laterally towards the lateral aspect. And further laterally, we find the sensory nuclei. These sensory columns can further subdivide into those receiving somatic sensory information positioned more laterally and those receiving visceral sensory information which are located slightly more medially within the sensory area. This longitudinal arrangement into functional columns is not just an anatomical curiosity. It reflects the developmental origins of these nuclei and the fundamental similarities in their function. During embryonic development, the neural tube, which gives rise to the brainstem, is organized in a way that dictates this columnar arrangement. This organization provides a very useful framework for understanding the location of specific cranial nerve nuclei within the brainstem. As we go through each cranial nerve, we will refer back to this basic columnar organization to help you visualize where their nuclei are situated and how their functions relate to their location. Let's now focus on the motor functional columns, those responsible for carrying efferent or motor signals away from the brainstem to control muscle activity. We have three main motor columns as you have seen in the previous slide, each with a distinct location and function. The most medial of the motor columns is the general somatic efferent or GSE column. As you have already discussed, somatic refers to skeletal muscle and efferent means carrying signals away from the central nervous system to cause action. The nuclei within this column are responsible for controlling muscles that move the eyes and the tongue. These include the oculomotor nucleus, located in the midbrain at the level of superior colliculus, and controlling most of the extraocular muscles responsible for eye movement, specifically the superior rectus, inferior rectus, medial rectus, and inferior oblique muscles. Next is the trochlear nucleus, which is positioned also in the midbrain, but at the level of the inferior colliculus, and that innervate the superior oblique muscle, which is another muscle that moves the eye. The next one will be the abducent nucleus found in the pons, controlling the lateral rectus muscle, which moves the eye laterally. And finally, the hypoglossal nucleus located in the medulla, which innervates the intrinsic and extrinsic muscles of the tongue, controlling its movements for speech and swallowing. So this medial column of general somatic efferent is primarily involved in controlling skeletal muscles derived from somites, specifically those moving the eyes and the tongue, allowing for precise and coordinated movements. Moving laterally is the branchial motor or special visceral efferent column, SVE. The nuclei here are involved in controlling muscles derived from the embryonic pharyngeal arches. These arches give rise to structures in the head and neck, and the muscles they innervate have unique functions related to these structures. These include the motor nucleus of trigeminal nerve in the pons, which innervates the muscles of mastication, allowing you to chew. The facial nucleus, which is positioned also in the pons, controlling the muscles of facial expression, enabling you to smile, frown, and perform a wide range of facial movements. And finally, the nucleus ambiguous, which extends through the medulla and contributes motor fibers to glossopharyngeal, vagus, and accessory nerves, innervating muscles of the pharynx, larynx, and upper esophagus, which are crucial for the swallowing and speech. And these fibers also supply the sternocleidomastoid and trapezius muscles, which control head movement and shoulder elevation via the cranial root of accessory nerve. The most lateral of motor columns is the general visceral efferent or GVE column. This column houses the parasympathetic motor nuclei. You have to remember that the parasympathetic system is part of the autonomic nervous system responsible for rest and digest functions. These nuclei control smooth muscle, cardiac muscle, and glands. These include the Edinger-Westphal nucleus, which is part of the oculomotor nerve complex in the midbrain and controls pupillary constriction, adjusting the size of the pupil in response to light, and lens accommodation, allowing you to focus on objects at different distances. In the pons, we identify superior salivatory nucleus, which contributes to salivation by innervating the submandibular and sublingual salivary glands. Further down in the medulla is the inferior salivatory nucleus, which also contributes to salivation, but through the glossopharyngeal nerve, innervating the parotid salivary gland. And finally, the dorsal motor nucleus of the vagus nerve, which is positioned also in the medulla, which provides parasympathetic information to a wide range of visceral organs, including the heart, the lungs, the digestive system, the slowing of heart rate, promoting digestion, and constricting bronchioles. Now, let's turn our attention to the sensory functional columns, which carry afferent or sensory information towards the brainstem. These are generally located more laterally within the brainstem, reflecting the general anatomical principle that sensory structures tend to be located dorsally and laterally. The first one among them is the general visceral afferent column or GVA. It receives sensory information from the viscera, meaning the internal organs. The main nucleus for this is the caudal part of the solitary nucleus or nucleus tractus solitarius, which receives input from glossopharyngeal and vagus nerves. This input includes a variety of sensations from internal organs, such as distension or stretching of the digestive tract, chemoreception, which is sensing changes in blood chemistry, and baroreception, which is sensing changes in blood pressure. These GVA neurons often have close anatomical and functional associations with the GVE neurons, which control motor output to the viscera, reflecting the importance of sensory feedback in regulating visceral functions and reflexes. The special visceral afferent or SVA column deals with the special senses of taste and smell. The rostral part of the solitary nucleus receives taste information from the facial, glossopharyngeal, and vagus nerves. This nucleus processes taste signals from taste buds on the tongue and other parts of oral cavity, allowing us to perceive different tastes like sweet, sour, salty, bitter, and umami. While smell is also a special visceral sense, the olfactory pathway from the olfactory epithelium in the nasal cavity projects directly to the olfactory bulb and then to the higher centers in the brain without a significant relay in the brainstem nuclei in the same way as taste. This is an important distinction to remember. The general somatic afferent column, that is the GSA, is responsible for receiving general sensation from the head and face. This include a wide range of sensory information from the skin, mucous membrane, and even some deeper structures. The primary nuclei for this are the sensory nuclei of trigeminal nerve, which are quite extensive and complex, reflecting the diverse sensory functions of this nerve. These nuclei include the chief sensory nucleus, also known as the principal sensory nucleus, which is primarily responsible for the fine touch and proprioception, allowing you to discern light touch and the position of your face. Then the spinal trigeminal nucleus, which processes pain and temperature sensations from the face and also receives some input from other cranial nerves. And finally, the mesencephalic nucleus, which is unique in that it contains the cell bodies of primary sensory neurons and it deals with proprioception from the muscles of mastication, allowing you to sense the position of your jaw and force of your bite, as well as proprioception from the temporomandibular joint. These nuclei extend through different levels of the brainstem from the midbrain to the medulla, forming a continuous sensory column, which is clearly visible in this diagram. Finally, the special somatic afferent or SSA column is dedicated to the special senses of vision, hearing, and balance. These senses have specialized sensory organs in the head. The vestibular nuclei, located in the pons and medulla, they receive information about balance and spatial orientation from the vestibular apparatus in the inner ear. These nuclei are crucial for maintaining posture and coordinating eye movements with the head movements. The cochlear nuclei, also located in pons and medulla, receive auditory information from the cochlea in the inner ear. These nuclei process sound information, allowing us to perceive different frequencies and intensities. And regarding the vision, although the primary processing of visual information occurs in the diencephalon and cerebral cortex, the nucleus of the optic tract in the midbrain is functionally related to the processing of visual reflexes, such as the pupillary light reflex and coordinating eye movements in response to visual stimuli. While the optic nerve itself originates in the retina and is technically an outgrowth of the diencephalon, its functional connections within the brainstem make it relevant to this discussion. We will continue with the functional component and nuclei of individual cranial nerves in the next video. Thank you for your patient listening.